Inside you, right now, are cells that carry a history of every significant infection you've ever defeated. They aren't memories in your brain, they are physical, living scars. Consider a single memory B cell. It is a veteran of a past war, carrying a unique genetic sequence on its surface, a molecular ghost of an enemy it once faced. This cell is not just remembering, it is the memory, a specific biological solution to a problem you already solved, perhaps years ago. How does one cell become such a perfect weapon, tailored to a single invader out of trillions of possibilities? The answer is a process of frantic, directed evolution that happens inside your lymph nodes. In structures called germinal centers, B cells that have seen an invader are pushed to mutate. Specifically, they mutate the genes that code for their surface antibodies at a rate 1 million times higher than the rest of your genome. This is called somatic hypermutation. It's not random chaos, it's a high-stakes competition. B cells produce slightly different antibodies, and only those that bind the enemy tighter are allowed to survive and divide. It's like forging millions of slightly different keys for a single complex lock, and then only keeping the ones that turn perfectly. Once this microscopic tournament ends, the winners, the B cells with the most effective antibodies, don't just disappear. The best of the best differentiate into long-lived memory B cells. They retreat from the bloodstream and find quiet niches, primarily within your bone marrow. Here they can persist for decades, metabolically silent. They are not actively fighting or dividing, they are waiting. This state, known as quiescence, is a biological marvel, allowing a cell to preserve its highly specialized state for almost an entire human lifetime, a sentinel asleep at its post. The signal is the return of the original enemy. When that same virus or bacterium enters your body again, even years later, these memory cells recognize it instantly. The activation is incredibly rapid, bypassing the slow initial stages of a first-time immune response. The memory B cell begins to divide explosively, a process called clonal expansion, creating a massive army of identical copies. Many of these copies then transform into plasma cells, which are essentially biological factories dedicated to one task: pumping out staggering quantities of the exact high-affinity antibody that won the evolutionary race years ago. This is not a search for a new solution, it is the execution of a pre-existing one. A primary infection might take a week or two to build a proper antibody defense. A secondary response, thanks to these memory cells, can flood your system with superior antibodies in just a day or two. This entire elegant system of directed evolution and long-term cellular storage is precisely what we are hijacking with vaccines. A vaccine introduces a piece of the enemy, a spike protein, a deactivated virus without the threat of disease. It's a training exercise for your immune system, tricking it into running that whole process: the germinal center competition, the selection of elite B cells, and the creation of a standing army of long-lived sentinels. The protection you get isn't from the vaccine itself, it's from the living memory your own body creates in response to it. Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.